Thank you for having me there. And what I'm trying to do is give you a brief background on how this methylation-based diagnosis thing works. What's the idea behind it? This shall take about 20 minutes. And then I have a couple of cases, if you're interested to discuss. This can be something which I would wish to be quite interactive, and maybe Carla, you can moderate this. Let me start with the background. So yeah, we have this WHO. In fact, 3 of the panelists here participating are just very much involved in generating the next version of the classification. And I think we can assure you that methylation-based diagnosis in the next version of WHO will play a significant role. As you can see from this graph, I'm trying to simplify. So what you see here is the fate of a cell throughout differentiation. Cells start out with a highly proliferative potential, low differentiation, and then they progress and move along some path and end up at a more differentiated cell with a slower proliferation. And this entire path is governed by constant switching on and off of genes. And this, as we know, to a major extent is regulated by promoters, and by the methylation status of promoters. So along this path, a cell will always have a very characteristic pattern of methylation, a global and especially a global promoter methylation pattern. And during this path, something can happen, something bad. Here, since most of us, I assume, are somehow attached to neuropathology, I took an example, an oligodendroglioma. This precursor cell, somewhere on its path, may have a mutation, IDH1 or IDH2, 1p19q codel, maybe some other mutations, early mutations, CIC mutations, p-TERT mutations, and maybe later on a CDKN2 AB homozygous deletion. All typical lesions in oligodendroglioma. What this must implicate is that this normal path of differentiation is interrupted. So the cell does not proceed to its final differentiation status. And that's the crucial point. We have here, at that time of tumor initiation, we have a distinct methylation profile, and just based on experience, this methylation profile is more or less, with minor alterations, but at large carried through the entire tumor evolution. That means if we look at the early tumor, or maybe here at a later tumor, and we look at the methylation profile, we are looking into the cradle, into the differentiation status of the tumor originating cells. And for this reason, this approach is really useful for the classification of tumors. And as you already can deduct, since there is not a major change throughout progression, it obviously is not very powerful in grading of tumors. There are some possibilities, but overall the major strength is in classification. The tool we are using is, in this case, is the well-known 850K chips, but I can assure you that it also works if you do methylome sequencing. Everything I'm telling you later on, everything would work also with methylome sequencing, requiring a couple of alterations to the algorithms, but it would work. So we like that chip because it has one great advantage for us, or two, it's extremely robust, and it works wonderful with DNA from formalin-fixed paraffin tissue. These are, for us in neuropathology, really major advantages. We analyzed tumors, actually a lot of tumors. And based on this large series, which we analyzed over the last couple of years, we initiated analysis such as this TSNE analysis, a two-dimensional reduction. What you see here is a landscape, and you see these islands of different color. And each of these islands really corresponds usually to a quite well-established tumor entity, which we already know from our morphological analysis. So what I find crucial in this depiction is you don't have a starry sky. It's not like the dots are randomly distributed and colors are randomly distributed, but you really see islands, focused islands of isolated colors, meaning that for example here here are all the glioblastomas, or here's the glioblastoma with the G34 mutation in the H3 gene, or here's the diffuse midline glioma with the K27 gene, and here's oligodendroglioma and astrocytoma, the IDH mutant tumors. So this distribution, of course, would be a wonderful basis for setting up a classification on top of this. And we did this by devising an algorithm. This algorithm has the major purpose to get a rapid analysis of a tumor sample, because if we take a tumor sample and we calculate it into this matrix, this is a lot of calculation power, of course. And the algorithm can do this in a fraction of a second. The algorithm works. It's based on a couple of thousand, well, on 10,000 decision trees. The decision trees have branch sites, and depending on the methylation status of a distinct CPG site, which is encoded by the chip, you either go to the left or the right until you come to some kind of a terminal node, which gives a ballot, a vote for a distinct tumor. Could be glioblastoma here of the RTK3 type, could also be meningioma. Well, after you have run your data set through this algorithm, you're coming up with a list depicted here, and just counts. For example, in this instance, the IDH mutant astrocytoma received 6,238 votes of the 10,000 decision trees. Next is oligodendrogliomas, and most of the other tumors, as you see here, have next to nothing. So the class prediction, in fact, is then something which is a decision by a majority vote. There's a second very important and completely independent way of looking at this data, and this is by using them to generate a copy number variation plot. So you can read this like this. Here's chromosome 1, 2, 3. Here's chromosome 22. In the middle, would be a balanced chromosomal status. And here, in this case, we have losses on chromosome 3, and we have here a gain on chromosome 12 on the short arm, a couple of losses here. So this is very interesting because several tumors have highly characteristic copy number profiles. I'll show you the oligodendroglioma later, or also the glioblastoma has a typical profile. And important is that this is entirely independent from the methylation-based classifier output. So these two do not affect each other, and therefore they can be very well used as two sides of information, either contradicting or supporting each other. It works very nice in Heidelberg, and in order to get this working also in your vicinity, we set up a web page. Actually, my colleague Daniel Schrimpf did this. If you want to take a note, this would be a good note: www.molecularneuropathology.org. So this web page explains you, if you run through it, what we are doing, how the reference sets look, what the typical characteristic of the reference sets are. But it also can serve for more. After you have logged in, after you have signed in, there are little information we need, like your email address and your institution. After you've signed in, you can upload data. You can generate the IDAT files in your laboratory, and then if you wish, you can simply upload them. We hope that you will tick this little box, which would allow us to keep the data, of course, in an anonymous way, to further learn the classifier. And you send it off, and it takes a couple of seconds, and you get an email to a registered address with the following information. Here, a couple of quality controls. Then you receive the output of the classifier. In this case, a glioblastoma with a very high calibrated score. The calibrated score is very important. if you receive an output with a calibrated score lower than 0.9, we strongly advocate not to use this as a prediction. So 0.9 is a threshold. Everything above 0.9 is good. Below 0.9 should be ignored. And then you also, in addition, receive this copy number profile. And you can use this for your scientific or diagnostic evaluations. And this is for free. Doesn't cost anything. This graph gives you the development of the use, how frequently this website is used. And you see we started in 2016 in the end, and we have a constant increase of uploads. Currently we are about in the range of probably 1,200 to 1,500 uploads per month. It's a lot. And here you see that most of the uploads still come from Europe, but a lot from the United States, but also South America, Asia, Australia. So this is really a worldwide network which is established there. So one great benefit which from it, something very exciting, which we realized actually only after we've set this up, is that this really is a resource for characterizing and identifying novel tumor entities. By now, we have approximately 80,000 data sets from tumors. This is roughly twice as much than a neuropathologist sees in his lifetime, and probably a couple of numbers higher, the number of cases a neuropathologist can remember. This provides us with fantastic opportunities. And I'll just give you a glimpse into the future of these classifiers. This is the system we are operating now. It's the version 11.4, which is based on 2,800 reference samples. Right now, we are preparing the release of a novel of the version 12, which is based on 6,000 tumors. And what you see here is the same color codes, these aisles of tumors. But what is new in this depiction is you see these little gray islands there, novel gray islands. I have a magnification. And again, these gray islands usually come in clusters. They are not randomly distributed. And we strongly think that these gray islands here, that they represent either molecular subgroups of established entities or even completely novel entities. So over the last 2-3 years, we have identified approximately 20 to 30 novel tumor subgroups and entities. And as you can see, as you can estimate, there is another 40, 50 to come within the next 2 or 3 years. And this is something which is virtually not possible without a tool like this, which allows the analysis, large-scale analysis of tumors, and look for likeness. So what we have here is probably tumor entities where we only know one thing. They share a common methylation profile. And from here we have to go and make contact to all our colleagues who submitted these cases and ask them whether they have clinical information and so on. And that is the basis of starting to characterize these dots and probably identifying several novel tumor entities. Works very nice with brain tumors, but in principle, this should also work with other tumors. And it does, I tell you. This is an unpublished data set. In fact, it's just in consideration for publication. And this shows you a TSNE, which is at the basis for a classifier for sarcomas. And similar to brain tumors, you see here roughly 60 islands, gastrointestinal stromal tumor, chordoma, leiomyosarcoma, and so on. And it's the same story. It works synovial sarcoma. And based on this matrix, we have devised, similar to the brain tumor classifier, a sarcoma classifier. And this works exactly the same way as a brain tumor classifier. The only thing you want to change is it's not molecular neuropathology.org, it's molecular-sarcoma-pathology.org. And you can also, after you have signed in with the same access, you can upload sarcoma files, IDET files, and you have a good chance of receiving a useful answer for this. It's not as advanced yet, and it's not as mature as the brain tumor classifier, but we think it already operates quite well. Same thing, upload, and this is the readout you receive. Well, the big one, of course, is would it work with carcinoma? And again, a major consortia, and I think also a lot of other groups are already starting with this. And this is just a very, very rough and dirty little TSNE which I prepared, which shows that also epithelial tumors tend to cluster in groups. And in fact, what you see here in brown color, this is all cancer of unknown primary. And what I think is, of course, this is a highly useful and very rapid tool of getting some idea where the cancer of unknown primary could come from. For example, these ones, I think they probably are of pancreatic origin, and this probably are from lung cancer, and so on. So this is just here to demonstrate that this may be also a highly interesting approach to be pursued for epithelial tumors. Well, with this, I'm coming to the end with the introduction, and I would like to draw your attention to one paper which you may not know, but which I would suggest as a guideline, because in this paper we have compiled our experience, the information we consider very necessary to use the classifier, and also have discussed a lot of pitfalls which we ended up with, and which quite took some time for us to find out how to deal with it. So this is like a guideline for using the classifier, and you can take a look at this, and I think it's helpful. This is my final slide. This is to state that, of course, this is not my own work, but it's work of a major consortium from two groups in Heidelberg, but also internationally contributing and helping with this enterprise. Thank you very much for the first part.
Okay, so I'm going to do this really quickly. I only have a few slides, but just as an overview, I'm just going to go how we've been bringing the methylation arrays in-house, how we've gotten clinical buy-in, what the workflow is like. I'm going to talk about everything but the validation, and the reason for that is because we only got 2 runs done because we started this in February and then COVID hit, and we've just been stalled out completely since then. And that's why ordering and reporting has a star on it, because this is what we plan to do and not what we've actually done yet. Convincing the clinicians is a super important part, and I think that it's something that can help alleviate some of the politics side of bringing on a new assay. Really recommend targeting neurosarcoma first because of the already really well-developed classification algorithms that have been spoken about today. From a billing standpoint, if you run this as a diagnostic assist in the beginning, you can just eat the cost because it's a DRG on a lot of these patients because they're inpatients, and it's almost better to eat it at first, prove utility, and then move it into a paying test later. Obviously, tumor boards are ideal presentation mechanisms, so we have a monthly molecular tumor board that's actually kind of been postponed too because of COVID, but where this data was presented and utilized. And I think that it's really important that you don't forget bringing along the anatomic pathologist side. Like, I'm a molecular only pathologist, but bringing along the anatomic pathologist, because this is something that's going to theoretically change a diagnosis that they've made, that can theoretically supplant some of the super high revenue generating IHC and other assays. So this can be viewed as a threat in some ways, both in a professional sense and as a billing sense. But I think that framing this as a diagnostic assist tool, talking with them, and then always, if there's any sort of discrepancies in any findings, bringing that to them before obviously informing the clinicians. The other reason for neurosarcoma first too is that usually you have a ton of tissue and it's a high tumor cellularity. So those two things are kind of great for this assay to begin with, and it's one of the reasons that I've been thinking that I obviously want to try this on TUOs as much as possible, but many times those are very, very scanty samples, and so it's something that needs a little further refinement still. So from a workflow perspective, this kits come in sets of 2 chips. So if you're trying to make a clinical workflow, each of these chips can run they have 8 slots in them, but depending on how many controls you put on a chip, there can be anywhere from 6 to 8 samples. So Cedars is really, really fussy about the number of controls that they run on a chip, even with all of the evidence that you can run these sort of on a delayed or maybe sometimes even monthly schedule. So we actually only get 6 clinical samples per chip. The thing is, if you have a low throughput volume, so like say you're only running 6 samples a week, you don't want to batch this into every other week because your turnaround time is going to be abysmal. So what you have to do is you're going to have to actually aliquot out the reagents into sub-aliquots to be able to do that, because they're kind of set up. The kits are kind of set up so that it's 2 runs worth of thing. It can be run on the iScan or the NextSeq 550, so not the new NextSeqs and not the old NextSeqs. And the NextSeqs have to be relatively recent. Otherwise, you need an in-service to upgrade some of the software. On the NextSeq, we happen to have a NextSeq, and because our throughput isn't super high, obviously we're talking about one run a week, 1 or 2 runs a week. You really don't need the higher throughput of the iScan. Assay takes 3 to 4 days, and it can be quite tech intensive in the sense of time. So automation is really critical for the final day. I'm going to go over that on the next. So this is actually, there's a day 0. And the day 0 is like the bisulfite conversion and everything like that. So that's happening before that. And there is automation that is available that's already pre-programmed. It's this TCAN Freedom Evo 150 Plus. Illumina sells it, or Illumina recommends it, and it's pre-programmed to do this assay. You need 2 of them if you want to fully automate. But if you're going to automate, I really recommend day 3 with the extension and the staining, because there's literally a step in there where a tech sits, pipettes, then waits like 15 minutes, and then does it again like 20 times. And so it's not just time consuming; it's annoyingly time consuming. And it's one of those things that get distracted easily, and they can't do anything else. So it's a perfect step for automation. We're actually putting it on a Hamilton because we have some room to do that for Hamiltons for our sequencing. So that's where I really recommend that. There are breakpoints. They're usually at the end of the days. So what ends up happening is that without automation, it's like a 4-day assay, but with full automation, it's like you save like a day, because you're able to skip a lot of those breakpoints. Obviously, robots cost money, and there has to be some expertise to actually do that programming and stuff. You don't end up buying something that's pre-programmed to do it. So this is how really we're planning on ordering and reporting. We're going to basically just bundle the order with CNS, with our in-house NGS for CNS tumor. So right now, every single CNS tumor gets a reflex NGS, and so we're just going to just add this on in the end. And we're going to actually report these out together. So we're going to take the report from the methylation classifier, and we're going to add a section into our NGS report that basically summarizes those findings, and include a overall molecular pathology interpretation that has the sequencing findings, the CNV findings, the methylation findings, everything kind of together to be reported at the same time. This will probably add a little bit of time to our turnaround time, because the methylation classifier will only be once a week, but in general, the brain tumors are a little more permissive on that. Obviously, all diagnostic discrepancy will be worked up with the spinal pathologist before we end up sending out these reports. So for charges, we're actually thinking about the cost, because we don't run MGMT promoter methylation in-house, and it's a huge cost to us. So the way we financially justify this is that it's actually going to be cheaper to run these methylation arrays than send out the MGMT promoter methylation outside. Now, I didn't tell the financial people that I could just set up a simple, really cheap MGMT promoter methylation assay, but that's fine. And so this will be a cost savings for our institution because of that. And as more clinical data is generated on the actual clinical utility of the assay, especially locally, we can begin to approach the payers. I love the idea of using that code for the array for neoplasia, but brief looking at MolDX submission requirements, it's likely that we're going to have to actually do an 81479 at first with a full technical verification and clinical validity submission. The problem with listing it as 81479 is that the other payers just like auto deny it. So you really have to go specifically to every one of your major payers. And if you're in a fragmented marketplace like Southern California is, it can be very difficult to reach out to every single one of them. But I think that it's just one of those things that over time, I'm going to echo, really does need its own code and probably needs its own interpretation code as well. Instead of just like the G code being listed. So those are my slides. If anyone has any questions, they're welcome.
I'm going to start off today just with a quick case presentation and a demonstration of the methylation classifier that we've set up for brain tumors at UCSF. This example is a 6-year-old boy who presented with headaches and nausea, was brought in by his parents, and was found to have an enhancing mass that was centered in the 4th ventricle in the posterior fossa, as you can see here. And histologically, this was a medulloblastoma and was of the desmoplastic nodular histologic type. And the disease entity medulloblastoma that looks like one of 2 or 3 different histologic subtypes under the microscope. There are actually 12 distinct epigenetic subgroups of this tumor associated with distinct genetic alterations and also distinct clinical outcomes. This was an example of a medulloblastoma that was run on our comprehensive targeted next-generation sequencing panel at UCSF that covers almost all of the known genetic alterations that currently happen in brain tumors. And the only thing that we were able to identify was some chromosomal copy number alterations in this case that were not specific for a particular subgroup of medulloblastoma. And the thing that's important to know is that many of the group 3 and group 4 medulloblastomas are driven by enhancer hijacking events that can't be reliably assessed by targeted NGS panels. And so really the best subclassification and prognostic information for the disease medulloblastoma is really provided by DNA methylation profiling at present. And so this is an example of a TS&E plot, and this patient's tumor is this black circle with an arrow pointing at it. And unfortunately, this patient's tumor falls within the group 3 medulloblastomas. This is a TS&E plot, which is a visual spatial representation of DNA methylation clustering, as we've discussed. But it is also classifying as a group 3 medulloblastoma based on random forest classification as well. And so those 2 methods are actually complementary in confirming DNA methylation profile and aligning with a specific entity, both either TS&E and UMAP plotting as well as random forest-based classification. And these are there's 4 main epigenetic subgroups of medulloblastoma. and this child's tumor belonged to group 3, which is shown here on this Kaplan-Meier plot in yellow, which is actually the most unfavorable prognostic subgroup and indicates a need for more aggressive radiation and chemotherapy and follow-up surveillance for this patient compared to one of the more intermediate risk subtypes, or the WNT one, which is the most favorable one. One other thing that I wanted to talk about today was the DNA quantity requirement as well as the relative tumor content requirement for reliable classification. And so what we've done here is we've taken DNA from a medulloblastoma that reliably clusters and reliably classifies using the DKFZ methylation classifier. And we ran methylation arrays using decreasing quantities of DNA input. And what you can see is when we run the recommended 250 nanograms of DNA, we get a very high calibrated score, 0.99, which is as high as it can be for the methylation class medulloblastoma sonic hedgehog activated subtype. And there's actually epigenetic subclasses that this one also matches with. This was an infantile tumor. And you can see that it also reliably matches with the methylation subclass of infantile SHHB as well. And when we went through decreasing DNA quantities, what you can see is that we were getting reliable methylation class prediction all the way down to 50 nanograms if you use 0.9 as a calibrated score cutoff as recommended. And you can even see down at 25 nanograms, we're getting the correct prediction, although it's below what we should be using as a good confidence score for clinical diagnosis. But I think this is a good indication for letting everyone know that, this is what the DNA quantity is required for doing these methylation arrays. And then one more slide. This is showing the relative tumor content requirement. And so what we did was we took DNA from this same patient's tumor, and we also extracted DNA from normal, uninvolved, non-neoplastic cerebellum from this patient. And we made mixes of the tumor DNA and the normal cerebellum DNA, and then ran these on methylation arrays using a total DNA quantity of 250 nanograms in all cases, but at relative different ratios. And what you can see is that when we go down to all the way down to 40% tumor content and 60% normal, we're still getting reliable classification with a calibrated score above 0.9. When we dip below 40% tumor content down to 30% or 20%, we're actually still getting an accurate prediction based on the methylation classification using random forest, but is now falling below this diagnostic threshold calibrated score of 0.9. One of the questions that we're still working to address is this going to be true of all tumor types and all admixed normal? If what we had was a mixture of an IDH mutant astrocytoma and cerebral cortex instead of medulloblastoma and normal cerebellum, is the tumor content requirement going to be different? Are tumors that are mixed with lymphocytes and plasma cells and microglia and macrophages, what is the tumor content required there? I think some of these are not well established, but I think that one of the important things is just having as high of tumor content as possible for the analysis is going to make it more robust. And so, carefully using a trained pathologist or neuropathologist, selecting the area of the tissue that's going to be used for analysis is an important facet of doing these methylation arrays and getting the most robust results out of them. And that's the end of the slides that I have. I'm happy to take any questions. I know we're kind of short on time, but at UCSF, we've been working on methylation arrays for about a year and a half now. We've been working in the neuro-oncology space, but have also been working on developing more of a pan-cancer neoplasia classifier, and working in multiple other organ systems as well. So I think that this is going to turn out to be a robust way of tumor classification, not just in neuro-oncology or sarcomas, but really across the full spectrum of human cancers, and really I think is the future of diagnostic pathology for cancer. That's actually how these 12 different the 12 different subtypes are actually defined is by methylation. So I would actually say it's impossible to do it without methylation. There are some genetic alterations that do tend to group with these specific subtypes, but I think that the most reliable and practical way of grouping and providing prognosis for medulloblastoma patients is using DNA methylation arrays at present. It's also possible to do it by transcriptional profiling, by RNA-seq, but the most robust and cost-efficient way at present is really these DNA methylation arrays.